So welcome on behalf of myself and Dr. Layla, my co-moderator. And our first speaker is Sami Bizzari, who's a database curator and researcher working at the Centre for Arab Genomic Studies, CAGS, the hosts of this wonderful conference. He graduated with a Master of Science in Human Genetics from the Imperial College, and at CAGS he works on contributing to the growth of the CTGA database. He authors publications focused on genetic disorders in the Arab world, and he also contributes to organising events such as this conference. So Sami's talk title is Revamped CTGA Database, Systematic Repository of Disease, Gene and Variant Bibliographic Data from the Arab World. Thanks, Sami. Looking forward to hearing your talk.
Bismillah, Assalamu alaikum. Thank you, Dr. Moon, in advance for introducing me. I'm glad to open the Genomics in Society module by showcasing the latest upgrade of our ethnic genetic database, the Catalogue for Transmission Genetics in Arabs, or CTGA. I'm part of the team organizing this conference, and so this talk has been recorded beforehand in the event that I get preoccupied. So here's a quick overview of the talk. I plan to briefly mention the need for Arab variant data and public databases and some of the resources currently contributing to this. For the main part, I will introduce CTGA, the kinds of data hosted in our database and our data sources. And finally, I will present a snapshot of our most recent major update, which involved a comprehensive survey of data from Lebanon derived from the literature up to the year 2020. And I'll conclude with our current focus and some of the prospects we're hoping for, inshallah. So the rate of data production on genetic disorders has grown tremendously over the last couple of decades. To process all this information, numerous databases were made that focus on clinically relevant genomic variants. And these resources can be roughly categorized into a few main types. There are the general variation databases, which describe variants in most genes with a relatively cursory or summarized description of phenotypes. There are the locus-specific databases, which number in the thousands and alternatively provide highly detailed variant observations for specific genes or set of genes. There are the resources that handle data from large-scale studies, which mainly focus on common variants and complex disease, and that also study population allele frequencies. And on the other hand, there are the national or ethnic genetic databases, which documents variants and phenotypes for specific populations. And such databases are essential for national diagnostic services, newborn screening, and basically informing healthcare professionals and the public of relatively common genetic disorders and alleles that are unique to the genetic architecture of the population. So a good example of this is the Finnish Disease Heritage database, which has many single gene disorders that are much more common in Finns due to founder effects. And in the Arab world, which I'll focus on, there's an established high general incidence of rare genetic disease, calling for much needed efforts to effectively catalog variant data in the region. So the Arab world is home to a diverse population with a heavy history of admixture and migration. In addition to the diverse gene pool, particular characteristics of the population include high age at conception, large families, marriage within tribal communities, and high overall rates of consanguinity, rates that far exceed those than that of Western countries, with an average reaching about half of all marriages. Because of this, Arab genomes exhibit unique characteristics. This particularly involves an increase in homozygous regions that are identical by descent in an individual's genome, and this is also known as the autozygome, and is thought to result in the higher burden of recessive disease. And clinicians have really relied on this enriched autozygome to identify and diagnose recessive disease gene associations through the use of homozygosity mapping, which basically narrows down candidate genes through a sort of process of elimination, focusing on these homozygous blocks in patients from inbred families. And this has also been exploited to more easily diagnose genetically heterogeneous conditions and find otherwise hard to pinpoint variants in patients. And in healthy autozygous individuals, the enrichment of loss-of-function homozygous mutations can help describe non-clinically relevant genes, which would otherwise be much more harder to study or identify in outbred populations. So altogether, the utility of studying such autozygous populations extends towards greatly enhancing the annotation of variants in established databases. And just to highlight this, there are published reports where variants catalogued as SNPs from healthy non-Arab individuals were later reclassified as pathogenic based on studies from the Middle East. And similarly, there were reports reclassifying hundreds of variants that were thought to be statistically deleterious in public databases like ClinVar and HGMD into benign variants. So databases that report on clinical interpretation, they need to be consistently updated to expedite sequence-based diagnosis. But unfortunately, there's a lack of representation and accessibility of Arab variant data. So for example, in the GWAS catalog, Middle Eastern data make up only 0.03% of their data set, which holds over 500 million samples, mainly from European ancestry. And similarly on Nomad, another widely used resource, which aggregates large-scale exome and genome sequencing data from various ancestries, there was virtually no representation of Middle Easterners in their version 2 release, which was their main data set until late 2019. Despite this, the issue of underrepresented Arab data has been addressed by many parties. So in 2005, CTGA launched as a public compendium of disorders in the Arab population, and the data originally presented as text summaries of published clinical and variant information arranged by country and literature references, similar to how OMIM report their main data. And following reviews of most of the GCC countries between 2005 and 2018, the database amassed over 2,500 records, and since 2018, we've cataloged roughly 1,000 more records. But before I get to the update of CTGA, I should mention some of the recent major public resources that are available today with Arab variant data. So, for example, in 2016, the Greater Middle East Variant Consortium collected West data from over 1,000 healthy individuals, and their variant data on allele frequency and zygosity is available through a browser and download file, and their data is split between geographical sections in the region. There's the Almina database in 2017, which holds variant data that combines the GME variant dataset with over 1,000 new samples, mainly involving genome and exome variant data from subpopulations in Qatar. And they similarly have a variant browser where you can pull the data. The same institute behind Almina also created the Disease Alleles in Arabs database, which holds a PubMed data mine of reports describing clinically relevant variants in the Arab world. And they mined roughly 1,100 articles hinting at the vast amount of relevant and peer-reviewed information that's available online. And in late 2020, Nomad's latest release has added thousands of samples to enrich ancestral diversity. This included over 150 Middle Eastern genomes, which is a great asset, although still relatively small to the other ancestries, which altogether make up 75,000 genomes in their dataset. Even more recently, an anthropological study of Arabs in the Middle East analyzed over 130 genomes from various populations, and they found millions of novel variants, and they highlighted ethnic-specific variants that underwent selection in the region. The biggest efforts, however, ongoing now are the national genome projects that are really setting the foundation for future research and genomic medicine in the region. As I'm sure you've heard from yesterday's talks, these projects are already achieving milestones, including here in UAE, Qatar, Egypt, Saudi, and Kuwait. So, for example, country-specific reference genomes have already been constructed from hundreds of nationals in some of the countries, providing some unique insights into variants in the region. And they've illustrated how using these reference genomes can improve the accuracy of subsequent genome analysis compared to using the standard reference genome that's used internationally. All right, so coming back to CTGA, our mission is to bridge the gap between the data that's published in the literature and the data that's available in public variant repositories. There's been a tremendous growth of publications in the field, and just to show this, a cursory search in PubMed for variant data in the Arab world shows an exponential growth of articles over time with all types of studies. And critically, many of the articles and follow-up supporting papers, they only get published due to describing new information, such as novel or extremely rare variants or novel phenotypes, which can go completely unnoticed by mainstream databases. So with CTGA, I will try to showcase how we systematically summarize the detailed data from the literature into an easy-to-access resource for clinicians and researchers in the region. So with our update, we decided to move away from our text-based descriptions of diseases and genes and address three main issues. We wanted to incorporate retrievable and universally described variant and phenotype data. And we wanted to show more data with a more concise presentation for the user. And from the curator side, we wanted to automate much of the data entry process. So the next few slides should give you a good grasp of what you can get from the database. There's a basic search, where you can input disease or gene names, their corresponding OMIM numbers, or you could search for the variant in HGVS terms, or with the dbSNP ID for the variant. There's an advanced search, which contains multiple filters that can be set and combined to create a specific query for each type of record. So, for example, under the variant tab, users could limit their search to specific countries or certain types of variants, like deletions or substitutions. And just as another example, for disease records, users could limit their search to specific phenotypes or disease classification. All right, so here's a typical disease record. The page starts off with a brief classification of the disorder, including items such as second names and mode of inheritance. And this is followed by a short description of the disease derived from OMIM. And you'd also find the gene that's associated with the disease here, which is linked to its own page on CTGA. And the main data in this record can be found in the epidemiology section. And in this section, in order to organize and contextualize the data here, we decided to orient the information based on anonymous subjects described from the studies. And the table is distributed across each Arab country. So each row in this table can represent one subject, or it can represent multiple subjects who share the same data grouped together. And examples of group subjects can be cohorts from association studies, or clinical studies reporting on many patients from an extended family who share the same clinical information. There are many studies that report summarily on unrelated patients, and when there's not enough unique information for each patient, we also group them together. So subject data have a couple of binary fields here, including family history with more than one affected individual, and parental consanguinity, considering all types of consanguinity. This is followed by a listing of specific clinical features for the subject that were translated into terms from the Human Phenotype Ontology. And following that, molecular details show the variant position in HGVS terms, as well as the zygosity and the segregation pattern that's reported in the study. The reference is also included here and is linked to PubMed, or if it's not on PubMed, it's linked to the article on the journal website. And from the same row, you can link to the associated specific variant page on CTGA and the subject record. And all tables on CTGA can be downloaded as an Excel file for further research. And data on subjects that are purely clinical or that do not have identifiable HGVS variant positions, these are added as text descriptions in the other report section. And this section basically has a link to the study followed by a short paragraph describing the key data. All right, so here's a look at the gene record. The gene record has a similar format to the disease record, so starting off with a summary and classification of the gene, as well as some gene and protein parameters. The epidemiology section here is oriented based on unique variants using the coding transcript by default, and the table is similarly split across the Arab countries. Some of the fields here include the chromosomal genomic variant position, as well as the established clinical significance, which is derived from ClinVar, and the associated conditions based on what we record here on CTGA. This is followed by the remaining HGVS expressions for the variant, as well as the dbSNP and ClinVar IDs, which link out to the respective databases. And selecting any of the variants in this table will send you to the corresponding variant record on CTGA. So finally, just a quick look at the variant record page. Here you'll find all the relative coordinates for the variant in HGVS expressions and the type of the variant. The epidemiology table here is oriented based on subjects, just like the disease record, however, with the addition of any healthy subjects that have molecular data reported in the study. So it could be healthy relatives of the proband or control groups in association studies. The allele count and allele frequency fields here are mainly there for describing group subjects. So an example here at the top is a report on a cohort of 16 unrelated patients who share the same clinical data and are homozygous for a CFTR variant. And just as another example, this is a case control study with obesity, where we could also derive the study-specific allele frequency for the particular variant. Other fields in the table include the clinical significance, which is specific to the reference in question and can differ from ClinVar, as well as the associated conditions and the reference. All right, so I'll just also show a glimpse of our data gathering and entry process. For the literature, we currently use PubMed and the Index Medicus Eastern Mediterranean region bibliographic databases. And recently, for our current survey of UAE data, we also started using Google Scholar, because Scholar also searches for keywords within the main text and not just the abstracts. So we're retrieving more articles which we would have otherwise missed from PubMed. The article selection process is manually created, and in the case that a subject's origin or country is not specified in the article, we contact the authors of the paper who typically share the details with us. We implemented automation to streamline filling in a big chunk of the data. So when we have our articles, we use a content management system to basically input and automate our data using APIs, which retrieves specific information from web sources. So for example, adding the PMIDs to our CMS would automate citations in the database. We then create the necessary disease and gene records where inputting the OMIM numbers would generate most of the data. So for the disease record, our sources include OMIM, OrphaNet, and WhoICD. And for our gene record, we use data from NCBI Gene, RefSeq, and UniProt to fill in all the gene and protein parameters. For the variant record, we input the mutation relative to the genomic transcript in order to automate the rest of the expressions, clinical significance, and IDs. And our APIs for this step include variant validator, specifically to retrieve the correct expressions, as well as ensemble and NCBI for the specific variant IDs and the clinical significance. And we also have a system in place to periodically recheck variants for which no IDs were retrieved initially through the API, in the case that they were updated later on dbSNP and ClinVar. And finally, the subject data is inputted manually, and at this stage, connecting the country, variant, and disease to the subject would automatically interlink all the data in the front end for the user. All right, so to showcase CTGA data, I'll just present an overview of our recent update of data from Lebanon. We started this project in 2019 with a PubMed search for biomedical literature referring to Lebanese subjects with variant data. And we ended up with over 800 articles from which we extracted over 600 disease records and 600 gene records. And we inputted over 1,300 unique variants with HGVS terms into CTGA. And these mutations mainly involved substitutions and small indels, and there were fewer reports of small haplotypes and microsatellite variants. And around 200 additional variants were added as text summaries to the other reports, and these mainly involved large copy number variations and large gene deletions and rearrangements in karyotype studies and linkage studies. So roughly 80% of the disease records that we catalog, they have a molecular diagnosis. However, the remaining records only describe purely clinical or prevalence data, which we added to the other report section. And many of these clinical studies were reported before 2010, which was roughly when sequencing-based and trio-based exome diagnosis entered the clinic. All right, so from the disorders we cataloged, most of the disorders were classified with an autosomal recessive mode of inheritance based on OMIM. And when considering subject-specific modes of inheritance, there was a similar trend with a higher representation of patients reported with recessive segregation patterns. So patients with homozygous or more rarely compound heterozygous mutations, and often connected to a healthy relative or the parent with the heterozygous variant. And the breakdown of the individual diseases based on classification showed a predominance of the congenital malformations and deformations class of disorder, and this is mainly attributed to most of the reports describing relatively rare monogenic syndromes. And similarly, monogenic metabolic and nervous system disorders were next most commonly reported. And from the disorders with known worldwide prevalence rates, roughly 80% would be considered rare that we cataloged, and most of the records we cataloged would be considered extremely rare with rates of less than 1 in 100,000 people. In fact, some extremely rare Mendelian disorders were reported in the largest number of non-group subjects relative to the other disorders we cataloged. So, for example, these included multiple autosomal dominant and recessive disorders across various classes of disease. And just to give a couple of examples, one article reported on spondyloepiphyseal dysplasia, which is a recessive musculoskeletal disorder reported in a large cohort of families. And that study identified 24 different pathogenic variants in one gene, including compound heterozygous variants in many patients. And over half of these variants were not described in ClinVar by the end of our survey. Just to contrast this, 3 articles reported on a subtype of autosomal dominant familial heart block, which was studied in just a single large consanguineous family spanning 5 generations. And this family was among other non-Arab families in studies which first mapped this disorder, and they found a heterozygous variant in 35 family members. BVVLS2 is another disorder first mapped in Lebanon, and this is a fatal motor neuron disorder for which, interestingly, some effective supplement therapy was discovered recently. And also notably, rare metabolic disorders were reported in large numbers of families. So, for example, congenital adrenal hyperplasia and the copper overload disorder, Wilson disease, were described in many families with several different variants. And as another example, one variant was described in patients from many families with a subtype of congenital lipodystrophy, which also involves severe diabetes mellitus. And this disorder was first mapped in Lebanon. So actually, at least 56 genetic disorders available in OMIM were first mapped in Lebanon, and these include many neuromuscular disorders, such as subtypes of Charcot-Marie tooth disease and various subtypes of recessive deafness. In addition to 45 of these first mapped disorders, an additional 108 were found to be only described in Lebanon with respect to the Arab world. And these included multiple subtypes of Fanconi anemia and other neuromuscular disorders, such as subtypes of congenital muscular dystrophies. And 13 of the disorders that are recorded in OMIM were found to be exclusive to Lebanon, and these involved recessive metabolic and immune system-related disorders and congenital syndromes. And some of these disorders describe just one clinical feature, and they were mapped through linkage studies. However, candidate genes were not yet identified. And finally, several articles describe disorders that were yet to be cataloged on OMIM, and these included 23 disorders, 11 of which we managed to catalog associated variants. So 5 of these disorders were novel diseases or allelic disorders with a unique clinical spectrum linked to substitution variants or small deletion variants. Other reports described chromosomal microdeletions and duplications that were associated with the syndromic conditions. And the reports that lacked molecular data, they mainly involved complex phenotypes with multiple congenital anomalies and intellectual disability segregating in the family, usually with more than one affected sibling. All right, and many genes were linked to multiple common, complex, and hereditary diseases, with variants mainly studied as potential risk factors or affecting drug response in case control studies. So here's just a cursory view of the most studied common disorders based on the number of alleles we cataloged for the group subjects. There were the typical disorders such as coronary heart disease, hypertension, and various cancers. And for the large drug response studies, multiple variants affecting warfarin drug response and anticoagulation were most prominently studied. However, case control studies reporting on type 2 diabetes involved the largest cohorts in our dataset. And common inherited disorders tied to the region include familial Mediterranean fever and more broadly beta thalassemia, studied in many patients with many variants. Hypercholesterolemia is another example, mainly attributed to the Lebanese allele that is prevalent in the region. And notably, specific subtypes of recessive deafness were also described in many subjects. Okay, so when comparing variants in CTGA against dbSNP and ClinVar, 150 of the variants entered with the HGVS terms were found to be absent from both databases. And 131 of these variants were reported to be pathogenic or likely pathogenic in relation to 109 disorders in the articles. And moreover, 274 variants were found to be on dbSNP but absent on ClinVar, and so lacking description of clinical significance. And considering that we're currently only hosting bibliographic data, this is a substantial amount of unique variant observations showing the utility of CTGA. And additionally, some variants in CTGA were found to exist on ClinVar with different clinical interpretations. So just an example, 2 SNPs for a myosin gene associated with recessive deafness were reported from healthy subjects on ClinVar. And for one of the variants, despite a report of it describing it as likely disease-causing and another as uncertain significance, a submitter labeled it as benign due to a high frequency of the variant in Ashkenazi Jews. And interestingly, the allele frequency was also high in the Middle Eastern data set on NOAA's latest release. However, no homozygotes were reported in either data set for either variant. But in the case of a Lebanese patient with recessive deafness, these 2 mutations in compound heterozygous state were determined to be the cause. This was from a study reanalyzing hundreds of exome diagnostic studies to improve methods, and it had other examples throughout. So I hope this gave you a good idea of what you can get from the database. We have many hopes and plans for CTGA. We are a small team at the moment, and so one of our major priorities is to set up and incentivize collaborations throughout the MENA region to ensure that data from each country is built up and then actively maintained by dedicated teams. So we're currently close to this goal for UAE, for example. Our survey is almost complete and we plan to keep it continually updated, inshallah. As for the database itself, our focus is on streamlining the most time-consuming parts of the data entry process, and this right now mainly involves getting the phenotype data for each subject. However, manual creation seems to be essential for this step, but there are some great online tools and potentially artificial intelligence down the line that we're looking to integrate in order to automate the process while maintaining quality control. And we also plan to expand our data search into other bibliographic databases. We hope to connect more resources into CTGA, such as implementing an automated variant classifier using ACMG guidelines to address previously undescribed variants. And we also want to incorporate variant allele frequency and zygosity data that's available in the public Arab variant databases. If you have published data that you want to contribute to CTGA, we have a submission portal online on our website where you can fill in a short survey, which we will then process and add to the database. So I would like to thank our great team, our past and present database curators, part-timers, and volunteers who really helped build and refine the database and who have helped organize events such as this. So thank you so much for listening, and I hope you find the rest of the talks beneficial. Assalamu alaikum.